it is my honor to introduce our speaker tonight, Dr. Kristen Ruegg. Kristen is an assistant professor at Colorado State University. Her research focuses on the development of innovative genetic-based tools to conserve migratory birds in the face of climate change and other stressors. She co-founded and co-directs the Bird Genoscape Project, which we will hear more about this evening. And I am personally very fortunate to have worked closely with Kristen in the past. My first field experience was misnetting swings and thrushes in the state of Washington with her. And my first job out of undergrad was working in a lab where I actually worked with Kristen on a couple of different projects, but that includes the early stages of the Bird Genoscape project. So when I started at SFBBO, I was also really excited to find out that our banding station does contribute feathers from some of the target species to the project. So I am very excited to welcome you, Kristen, and to hear more about the work you've been doing.
Excellent. Thanks, Serena. Okay, yeah, no, it's really fun to be here and see Serena in her new position. Thanks for the introduction. So yeah, today I was going to tell you a bit about the Bird Genoscape Project. In particular, I'm going to sort of talk about some of our core goals and objectives, which is using genomics to map population level migratory pathways in birds, and why that's important. And this was the work that Serena was part of, the very first sort of iteration. The next part, I thought I'd just give you a little bit of, updates or insight into how we're using some of the genomic data we're collecting for other on-the-ground conservation efforts, some of which are happening in the San Francisco Bay Area. So I thought that would be appropriate. And then a little bit about how we're connecting with education and outreach using the data. So I'll start with this first one, mapping population level migratory flyways. And we're interested in this because, at the foundation of all of this is this question of where do migratory birds found breeding in northern latitudes spend the non-breeding season. And we're motivated to understand this because of declines in North American avifauna. So this really elegant study by a group of scientists just back in 2019, summarizing a lot of the citizen science data that's come out over the last 70 years showed that, roughly 3 billion birds have been lost from North America since the 1970s. So at the sort of foundation of this is trying to understand the different stressors that birds are facing across the breeding, migratory, and wintering grounds, including habitat destruction, predation by house cats, avian diseases, and one of the main things my group works on is the potential current and future impact of global climate change. So where are birds most threatened? Those of us that think about migratory birds know that there are, many different places that they occupy throughout their annual cycle: breeding grounds, migratory stopover, and wintering grounds. And so what we're trying to do is identify these migratory connections, which is a first important step towards effective conservation. So if there's, habitat destruction on the wintering area of this yellow warbler, we know how that affects population trajectories on its respective breeding grounds. All right, so past approaches to link breeding and wintering areas have included, the North American bird banding program, which began in the 1920s and has been like a massive effort to track and monitor the movements of migratory birds. And with a lot of the larger bodied species, it's done really well. But with many of the smaller bodied songbirds, recapture can be really difficult. And in many cases, recapture rates in terms of long distance recaptures, so birds captured away from their initial site of capture and banding, are only 1 in 10,000. So this translates to a recapture rate of approximately 0.01%. More recently, there's been a number of really exciting approaches to have developed, including radar, telemetry, and GPS tracking. One of the coolest, I think, is the Birdcast program. So if you haven't checked out Birdcast before, you should Google it right now and check out some of the maps, because right now during migration is when it's really exciting to see these maps, which are basically weather radar stations all over the US, which are capturing sort of the noise, and the weather radar is actually, bird and bat movement across space. And so by taking the bird and bat movement and we can look at sort of broad front when big pushes of migration are coming through your area. So they'll actually predict, if you go to these maps, they predict, where and when, and in some cases, approximately how many birds are migrating through your area and in what direction. It's super cool. So this data provides really important insights at the level of sort of all birds. Then we have really exciting advances in, geolocators, GPS tracking, and MODIS, which, use a combination of, it's a combination of different tools, but essentially these provide really high resolution information, especially with satellite tags, at the level of an individual. So where does an individual bird move? How does it move across space on like a minute by minute basis? It's pretty amazing. But where the Bird Genoscape Project comes in is, how do we track birds at the population level? And why is this important? So why is tracking birds why is tracking migratory populations important? So to demonstrate this, I'll talk about the willow flycatcher. And the willow flycatcher is a widespread species that its breeding range is shown in green, and its wintering range is shown in purple in this map. Prior to this work, very little was known about population-specific migration patterns in the willow flycatcher. However, patterns of decline in the willow flycatcher are very population-specific. So this southwestern willow flycatcher, Extimus, is actually on the endangered species list. And so what we really want to know, what we really need is a population-specific map of migratory flyways so that we can understand where the southwestern willow flycatcher winters and then develop full annual cycle conservation strategies. So our approach to doing this has been the development of high-resolution genetic tags, which focus on a little bit of tissue at the base of the feather that contains the DNA. And with that DNA, we identify a unique genetic code that allows us to trace the breeding origin of migrant birds captured anywhere along the migratory trajectory, outside of the breeding range. So essentially what our method is an ancestry service for birds. So when you send, your spit off in a little vial to find out where your ancestors came from, this is essentially what we're doing when we analyze the DNA in the tip of a feather. We're able to understand the breeding origin of that individual based on its genetic code. Some advantages of this are that we can get at population-specific movement patterns from all birds as long as we have a feather. And feathers can be collected with pretty minimal impact. We can also use this methodology to identify individuals that, from carcasses where the birds were subject to collisions or maybe poaching or avian disease, or mass mortality events like, birds dropping from the sky for some unknown reasons, which unfortunately is happening with increasing frequency. We also have the potential, which is one of the favorite parts of the project, for connecting with biologists across the Western Hemisphere. So many of you who've worked at, the bird banding station with the San Francisco Bay Bird Observatory might have collected feathers for the project, which is really exciting. So thank you for your efforts. So I'm just going to go through the steps of how we create a genoscape. So the first is that we scan the genome for genetic variants. So if the genome is made up of A's, C's, T's, and G's, these nucleotides, we're scanning the genome for genetic variants that are representative of populations at finer spatial scales. And we do this in a number of different ways. But once we have the genetic data, we use that genetic data from across the breeding range to create what we call the genoscape. So each one of these black dots represents a location where we had breeding samples, and we scanned the genome of these individuals and identified these different populations that are color coded here in green, purple, blue, orange, red, yellow, and light blue. So based on the genetic data, we can identify a Pacific Northwest, an Interior West, an Eastern, a Southwest, a Kern, a Southern California, and a White Mountain genetically distinct group of the willow flycatcher. We then can use DNA from the feathers to identify migrants to populations. So this is where the sort of ancestry service comes into play, and where the collaboration with groups like San Francisco Bay Bird Observatory becomes critical. So these feather collections these efforts have been going on for 25 years. So this project was started by Tom Smith, who's at UCLA 25 years ago. And as a result, there's roughly 250,000 feathers from 100 species. And in many cases, they span a 25-year time series. And so these dots on the map represent locations where we have had banding stations that have contributed feathers to the project. So I feel like the map doesn't always do justice to just the number of partners that we have in the project. And I know you can't read all of these, but I just wanted to give a shout out to the fact that it is a very huge effort with contributions from hundreds of people every year. And so, yeah, and it couldn't be done without these contributions. So a lot of these are, different agencies, nonprofit, international nonprofit partners, individuals at US academic institutions, individuals at international academic institutions, primarily in, throughout Latin America, and then government partnerships, as well as hundreds of individual contributors across the Western Hemisphere. So we use the DNA then from the feathers to identify the breeding area of the migrants. So sorry, this is really where the ancestry service part comes in. So we take take the feather, extract the DNA. We develop a genotyping chip, it's called, for every species, and we run it through this machine called the fluidigm. And essentially what pops out at the other end is a set of genetic variants that distinguish populations. So for example, perhaps, AA is at high frequency in Alaska, TT is at high frequency in Texas, and AT is at high frequency in Oregon. Some advantages of this are that we can, as Serena knows, because she did this when she worked on the project, you can screen a lot of feather samples in a week, like roughly 800 feather samples in a week, and it works pretty well with low quantity DNA. So once we have screened all of the additional wintering and migratory samples, we can then connect, say, the Pacific Northwest breeding population. We can identify that it winters from Western Mexico down to the Panama and migrates along the Pacific Flyway. We can show this sort of purple dots down in starting in El Salvador and down into Ecuador, show that the Interior West population winters in those areas and migrates along the Pacific and Central Flyway. And then we have Eastern birds found wintering from Panama south to Ecuador and migrating along the Central Mississippi and Atlantic Flyway. And then most importantly, for this project in particular, we're able to identify the specific wintering areas of the endangered southwestern willow flycatcher in Nicaragua and Costa Rica. So we have been collaborating very broadly with partners. Again, many of the partners that I already listed. We're currently, either working on in-house or helping others with constructing genoscapes for about 50 different species of migratory birds. And for those of you, I'm not sure how many are familiar with the Road to Recovery effort, which has been a big effort to like prioritize conservation on a subset of species. These are just the R2R species that we've been working on recently. And we're always, looking for ways to expand or help others with, constructing genoscapes so that we can come up with sort of a consistent framework where we can, provide these maps and have them be the most useful to conservation efforts that we can. So one of the ways that's been really exciting for us to be able to communicate the connectivity and information that we've been generating to broad audiences has been working with Audubon's Migratory Bird Initiative, which just released last week. If you haven't seen it, you should check it out. So they just released the Migratory Bird Explorer website last week, which summarizes some genoscape data, a lot of the tracking data and banding data that's been collected over the last 30 years or so. And so you can go to a species and actually look up all the information, all the tracking information that's available for that species, which is truly an awesome resource. We've also been a co-author on the Smithsonian-based Migratory Bird Atlas, which will essentially be a book version of the Migratory Bird Explorer, summarizing a lot of the best data we have on migratory connections. Okay, so that's the sort of core part of the program, the mapping population level migratory pathways. I wanted to share a little bit about how we're connecting the Genoscape data to some ongoing conservation efforts, some of which are in your own area. So the first is we can use the same genomic information and the genoscapes at the foundation of this work to inform captive breeding and genetic rescue in species like the burrowing owl. So this work is really inspired by a collaboration with Lynn Trulio, who is at the Department of Environmental Studies at San Jose State University. And Lynn came and was, when I still lived in California, like over 10 years ago, I think, asked, can we use genoscapes to improve, captive breeding of Santa Clara Valley burrowing owls? And so we started by working with Lynn to create a burrowing owl genoscape. And so this is the genoscape shown in the left side of the screen here. And so what this revealed, interestingly, was that all migratory burrowing owls, which are shown in blue, basically have a very uniform range. And then all of the resident populations were genetically distinct. We also found that within all of the resident populations, we found higher levels of inbreeding. In particular, resident populations in the Santa Clara County were inbred and being threatened by habitat development or encroachment on their breeding areas by development. So we basically were able to use the same genomic data, the same genomic tools, which allowed us to construct the genoscape to identify and make recommendations on how to pair burrowing owls that were being brought into captivity and paired and then released. And so we use the genomic data to look at which individuals have the lowest level of inbreeding across their genome. And not only that, but which have contrasting patterns of inbreeding across their genome. So we can match individuals that don't have regions of the genome with the same signals of inbreeding. And that the idea is to maximize the genetic health of the burrowing owls in Santa Clara. So this has been going on for a few years now. So 5 pairs of burrowing owls were soft released into the wild in the spring of 2020, 4 pairs in the spring of 2021, and 7 pairs in the spring of 2022. And we're working on our next round right now. So overall, yeah, genoscapes provide a really high resolution option for informing captive breeding decisions. And they can also be used to inform genetic rescue. And this is the case where genetic rescue would be if individuals decided that inbreeding was too high in a certain area, they may bring burrowing owls from a remote location, not Santa Clara Valley, bring them in to basically import genetic diversity into the population. So this isn't happening now, but we are, we're involved in trying to provide the information if we can. Okay, another way we're using the genomic data that we're generating is to advise the U.S. Fish and Wildlife Service on ESA listing criteria needed for different species. So a lot of this work is in collaboration with Brenna Forrester, who works for the Division of Endangered Species with the U.S. Fish and Wildlife Service. And Brenna works on things called species status assessments, which are basically at the foundation of delivering the science necessary for informing the U.S. Endangered Species Act decision-making process. And so what we've been working with Brenna on is, can we integrate genomics into the U.S. Endangered Species Act? And we'll come back to the example that I brought at the beginning, which is the willow flycatcher. And so what I already sort of showed you at the beginning is that this genomic information, these genoscapes that we're creating, currently provide the highest resolution data available to support the ESA listing requirement that populations are genetically distinct. So for example, there was recently debate over whether to delist the southwestern willow flycatcher, and we could consult with individuals at the U.S. Fish and Wildlife Service, and share our data showing that the southwestern willow flycatcher is a genetically distinct population. Another way we're working with the U.S. Fish and Wildlife Service is to use we also use the sort of same genomic data to not only understand where the genetically distinct populations are, but to understand how populations are adapted to their local environment across geographic space. And I don't have a lot of time to go into this, but I'm happy to answer questions later. Essentially, what we do is we look at the relationship or the patterns of local adaptation across the range now, and then using models of future climate change, we can predict which populations will have the hardest time adapting to changing climate conditions. And so basically, genoscapes also provide a framework for predicting the capacity of populations to adapt to future climate change. So overall, this genoscape data provides a really high resolution option for both informing current genetic boundaries needed for ESA listing and for informing future adaptive capacity of populations to changing climate conditions. All right, the last sort of vignette I wanted to go over is the use of genoscape data to assess the impacts of wind and solar energy development on migratory birds. And this has been a collaboration with a lot of different agency and industry partners, many at the U.S. Fish and Wildlife Service, the U.S. Geological Survey, and then a variety of individuals within a group called the Avian Solar Working Group, which is, a lot of these individuals are part of the Avian Solar Working Group, and it's a sort of a consortium of companies and academics and organizations attempting to understand how birds interact with solar facilities. So what they came to us to ask is, can we use genoscapes to identify populations impacted by wind and solar energy development? So I will take one that Serena is very familiar with because she contributed to the original genoscape for the Wilson's warbler. In the Wilson's warbler, we'd identified 6 genetically distinct populations shown in the different colors. And then if you sort of zoom in on California with the pie charts on this map representing the location of different facilities of either wind or solar facilities where carcasses have been collected, we can genetically ID the carcasses, and we can assess which populations are being impacted. Is it populations from, the largest, our populations that are, declining being impacted, our populations that are remaining stable being impacted, and what sort of, how does that break down when we look at the population level impacts, and how does that sort of change our understanding of the threats to migratory birds? We can also genetically ID birds that are migrating through an area, which could help with mitigation. So these pie charts below, below the map. So Hollister Pass is a wind facility. And, we did sampling at a banding station near Hollister Pass, and then genotype birds as they were coming through to show when different genetically distinct populations migrate through that area. And this may help with, identifying when particularly vulnerable populations may be coming through an area, which may help with timing mitigation efforts. So overall, genoscapes provide a high resolution method for identifying the origin of carcasses impacted by wind and solar development, and can also be used to target the time of year when vulnerable populations are at risk. All right, so in the last few minutes, I will talk a little bit about our education and outreach activities, which are super fun. So we have been working for about 4 years now, maybe 5, with a company called Days Edge Productions. And this work was funded by the National Geographic to connect how Genoscape data can be used to connect the efforts of on the ground efforts in like the Kern River Valley with willow flycatchers led by Mary Whitfield, with those of individuals on the respective breeding areas in Costa Rica. And these have been a series of short documentaries, one of which, there's been a number, and they had different sort of audiences. One was a 15-minute documentary that that Days Edge put together that was hosted by National Geographic and did super well in the whole film festival circuit. There's another version that was for National Geographic Education. And so this is called Tales of a Feather. And the cool thing about working with Nat Geo Education is that they will provide curriculum materials for teachers and target the videos towards a certain age group. So that's been a really fun project. We also just finished doing the filming last summer for a Spanish language first documentary, which is being, again, produced by Days Edge Productions, but will be distributed by HHMI and through their biointeractive website, which again provides educational materials for high school and college level classrooms. And so one of the cool things about this is we got to feature, Latin American partners and film them in Spanish with English subtitles. And so we've been using these videos in our local outreach efforts here in Fort Collins. And so it's been a really fun project. So we had a grant from the National Science Foundation to support outreach with local Title I schools. In Colorado, a Title I school is a high proportion of economically disadvantaged students. And so the first day of what we call Bird Camp is in the classroom, where they watch the National Geographic video and learn about the project. And then we're actually doing it this week. So this Thursday is our first Bird Camp of this year. We bring the students out to the CSU Environmental Learning Center, where we host a variety of activities that allow the students to go deeper into the materials that they learned about in their classroom in the video. So like using DNA to track birds, learning about threats along the migratory pathway, bird banding, things like that. It's really, really fun. We also run international outreach efforts, mostly in the form of, education, bioinformatics, and genomics workshops. But they're also fantastic partner building at building, meeting lots of people and creating partners of individuals interested in working together on these issues. So we hosted our first in 2018 in Mexico City. Our second was hosted in Merida, Mexico, in the Yucatan Peninsula in January. And then we'll be hosting another one, not this year, but the following year in Baja, California. Okay, so today I've told you a bit about the core goals of the Bird Genoscape Project, which is to map population level migratory flyways. I told you about using the same genomic data to connect to on-the-ground conservation efforts for migratory birds, and also how we use some of the materials we generate to run education and outreach efforts. And with that, I would like to thank our funders, for without whom none of this happens. And yeah, so I am happy to take any questions.
Awesome. Thank you so much, Kristen. That was so fascinating. It's really fun for me to hear about how the Bird Genoscape Project has grown and expanded so much in the last 8 to 10 years since I have been on it. So I really loved hearing your presentation.
Excellent. I'm glad.
Yeah. So as a reminder for everybody, if you have any questions at all, go ahead and enter those in the chat and I will be reading those as they come in. And I did see a question come from Jan. So Jan asks, for willow flycatcher and Wilson's warbler, you identified a handful of distinct populations. Is that the end of the story, or is it possible that future DNA technology and software might lead to even finer level population distinctions?
Yeah, that's a really good question. I mean, I think at this point, we're actually sequencing the entire genome. So from a, just looking at genetic variance standpoint, there's not, we could get higher resolution whole genome data and it might help a little bit, but I think we're reaching the end of the resolution from genetic variance standpoint. Now there's something called structural variance, which is maybe more information than you want, but it's things like, in DNA, when it replicates, all sorts of things happen. There can be like translocations and, missing pieces, and all of those are other kind of, they contain other information. So it's possible that you could layer on top structural variance. Another thing that I did not go into in this talk, because I was sort of focused on just talking about why understanding things at the level of the population is important, is that we can, with our genomic data, actually do individual-based assignments. So we can take a single individual and assign it not just to a population, but to a particular spot on the breeding range. And that comes with, confidence intervals around it. But the more genetic data we have on the breeding range, the finer scale our ability to identify the specific breeding origin of an individual becomes. So I'd say for the Genoscape project, it would be more filling in the sampling gaps would be a better use of our effort than maybe adding more genomic data, which we're getting pretty close to the end.
Not to mention, you have so many species to work on.
Right, exactly. And, there's a big push, especially with a lot of our main funders, wanting us to do this for as many species as we can. So, yeah.
I mean, that's a nice problem to have, I guess.
Yeah.
All right, so our next question is from Rebecca. Can you say a word about how you collect the feathers from such a large number of species over such a huge geographic range?
Yes, I don't collect any of them pretty much. That's where we rely on these partnerships. So, bird monitoring stations like the San Francisco Bay Bird Observatory have been running all over the Western Hemisphere for decades. I mean, we're lucky in North America to have a lot of, especially like things like SFBBO, like that have been running for multiple years, and where they're able to maintain the funding to keep going. Those are, critical to efforts like this. And there's a lot of them. There's some that are more, ephemeral, primarily because they just don't have the funding to keep that sort of monitoring going. But that's how we are able to get the feathers. We collaborate with these partners, and the partners are the one doing the work on the ground collecting the feathers.
All right, and then we had another question from Patty directed to us at SFBBO if we have a MODIS tower, and we do. Katie is our science director and responded that we do have a MODIS tower at our bird banding station, and we'll soon put up 2 more in the South Bay. So the next question is, given that funds for conservation are limited, is it more important to save an endangered subspecies that is adapted to a specific region, like the subspecies of willow flycatcher, or to focus on the more populous, genetically diverse subspecies and hope they adapt someday? How do you make that choice?
Yes, that's a really, really good question. I don't make the laws, but like the moment, and I would actually totally agree. I mean, I don't know if that's what you were saying, but I at least agree that there's a lot of validity to the idea that we might want to start thinking about conserving areas, populations where there's already high adaptive capacity. But on the other hand, I think it's hard. Yeah, I mean, but the whole legal system is sort of set up around providing resources for saving things that have been listed. But what we're trying to do with the Bird Genoscape Project, and this is where I kind of, the R2R effort I found a little frustrating, because a lot of that was funneling all of our resources on these populations that have really narrow distributions and are already struggling. And, I agree with whoever wrote that in. Like, I feel like we need to be looking across, at all birds, ones that have common, more abundant populations and some that are declining, in order to more deeply understand what the stressors are, and then have more capacity for maybe, putting our efforts towards things that aren't already really hit hard. I don't know, but that's a good question. I don't make the laws though.
Yeah, that's a tough problem.
Yeah.
All right, and then keeping on that subject of conservation, you may have touched on this, but what results have you had so far on the impact of solar and wind power development on endangered migrants?
Yeah, very few of the individuals. So what we found, which is like not that surprising, I guess, it are that that the vast majority of impacts are on the most abundant populations. I mean, and that's just a matter of numbers. But like but it pans out like when you genetically. So for example, the willow flycatcher, we didn't have we have never had like a southwestern willow flycatcher pop up or anything on the endangered species list for which we have a genoscape. And the vast majority, for example, for the Wilson's warbler example I showed, were individuals from that large purple group, the Western boreal group, which is one of the most abundant groups of Wilson's warblers. So yeah, that's the general trend. Did that answer the question?
Yeah, I think so. All right, and then the next question, I'm curious about the southwestern willow flycatcher because I worked as a field assistant for Mary Whitfield one summer a few years ago. I noticed that you showed multiple colors within the range of the southwestern willow flycatcher, and I'm wondering if there are more distinct forms that are recognized and if smaller population units deserve conservation. A couple of the colors seem to be very restricted in range.
Yeah, I know. The willow flycatcher is really, really interesting. And I have a postdoc in the group right now that is, working on the willow flycatcher. We did find that like San Diego seemed like distinct from the rest of the Southwest. And also there was like the Kern and the White Mountains. And we think what might be going on there is really high levels of inbreeding in those areas. Because like, for example, the Kern is, at one point, I think there were only a few breeding populations left. And when you have inbreeding like that, it can cause like extreme genetic drift, essentially. Like it can really sort of change, it can change basically because of the inbreeding, and it can change the genetic signature. And the same thing with the White Mountains. It's like this high altitude mountaintop population, and we think it's probably very restricted. And so its breeding is probably very restricted, not a lot of gene flow. The San Diego is really interesting. And you have to wait for Sheila's paper for that one. But like what it actually seems like, a little teaser, is that San Diego seems to be a result, like we actually sequenced historical San Diego populations, individuals from the early 1900s. And so historical San Diego was very genetically distinct from any population now. And when we had the historical like marker of what it used to look like, you can see that present San Diego seems to be like a hybrid between Southwest and Pacific Northwest, and the historical San Diego. So it seems like there's been actually some mixture of individuals in that population. And we hypothesize that happened after the steep population declines in the early 1900s, but that's just a guess.
Oh, that's so cool. So interesting. All right, the next question. Are there specific genes that are known to be determinants of the best migrators that may be common among different migratory species?
That is a great question. Does this person want to come do a PhD? Yeah, so we are definitely trying to look at that. What we have found so far, we've been sort of doing it on a species by species basis. So actually, San Francisco Bay Bird Observatory helped with this. So we did this in the common yellowthroat and are working up the results of that project right now. So that project was led by Luz Zamudio, who is one of our postdocs that is located in Mexico. And Luz compared resident and migrant forms of the common yellowthroat, Bay Area, resident versus migrants, another in Arizona, resident versus migrants. And then on the East Coast, she had like Florida and Georgia, resident versus residents, and then versus migrants. And so that's a cool one, because for those of you that don't know, common yellowthroats are the migrant common yellowthroats actually originated out of two different resident populations in Mexico. So they're not each other's closest relatives. It's made up, migrants across the US are made up of groups where their sister taxa is in Mexico, actually. So it's super cool. So we have this really neat system where we have these two closely related groups with different evolutionary origins. And then we have the migrant resident comparison, and we're comparing the genes that are popping up as defining, the difference between migrants and residents across these multiple transects. And even within a species, there are definitely similarities, but there's also differences. So then we're kind of the plan is to expand this, not just to beyond common yellow throat. We've also done this in American kestrels, identified genes linked to timing. And, eventually we would like to do it for a lot of different of our migratory birds and see, if any of the regions pop up as similar. But anyway, good question.
Oh, that's amazing. And then a few more questions here. So why feathers rather than some other method of getting DNA like swabs or other methods? Is it that you were looking for something working on both dead and alive birds? Have you been getting DNA from museum specimens?
Yes. So feathers are, swabs, I think, would be pretty tough. The nice thing about a feather is the part of the feather that we use, as Serena knows, is the tip of the feather where there's the attachment site to the bird, where there's a little bit of skin. So it actually provides a little, just a tiny bit of skin. The other reason why feathers is typically birds are very familiar with dropping feathers. They will lose feathers quite regularly, and then they can well, you guys know this. I mean, they will regrow feathers. And so it felt like a less invasive way. Now, swabs could possible, but my guess as a geneticist is that it would increase the effort required to actually get the DNA needed from the swab. I don't know where you would take the swab. I imagine it would be pretty dirty. It just sounds like a bit of a nightmare. Was that the whole question, Serena? I forgot. I don't know. You've had to extract thousands of these. Do you want to be doing swabs or feathers?
I don't know. I'd have to try it out, I guess.
Yeah.
Yeah. And then the other parts of the question were you looking for some things working in both dead and alive birds? And have you been getting DNA from museum specimens as well?
Yeah. So dead and alive. Yes. So that's the carcasses from the solar sites are, dead. And mostly they're from live birds. The vast majority are from live birds. And yes, I told you about that study Sheila's doing. We also have been collaborating with John McCormick's lab at Occidental College, who runs or helps operate the Bear Divide Migratory Station, which is another one in the LA area. And they have the Moore Lab collection, which is these amazing treasure trove of samples from the 1940s and 50s from across Mexico. So we've been working with John to investigate, have patterns of migratory connectivity changed over time. So stay tuned for that. That is coming out as well.
Man, so many different
I know.
projects. So cool. Another question, do you include molt migration stopover sites in these data sets as well?
Great question. So yeah, we've been working with a bunch of individuals on the painted bunting, and painted buntings are becoming kind of a classic molt migrant species. We don't particularly, so what we did, so we don't we can't really get at the molting location with our methods, because our methods tell you where the birds breed, not where they molt. But if you capture the birds on their molting area, then we can say which breeders are doing the molt migrant. So that's been part of what we did. But in particular, we've been working with a postdoc named Andrea Cantina, who, Andrea, yeah. So Andrea was interested in the genetic basis of molt. And so we actually basically were able to use isotopes. Oh my God, this is complicated. Yes, we've been able to designate which individuals were molt migrants versus which ones were ones that molted on their breeding area. And then actually looked at the genetic signatures, associated with molt migrant versus molting on the breeding area. Yeah, and I discovered a whole bunch of genes linked to molting. It's a super cool paper. It's in review now. Again, another project.
We'll have to stay tuned for that study to come out. Okay, so the last question I'm seeing here for now. So I've worked on the American Kestrel Project for 3.5 years now and have taken feather samples from plenty of kestrels for the Genoscape Project. From what I understand, the feathers weren't able to give us results at as high of a resolution as we were hoping for. I know some colleagues who have been collecting talon samples for genetic studies. Do you know of any future plans to try to tease out the differences between kestrel populations to help us determine the connectivity between the populations?
So the Kestrel Genoscape had an Alaska group, a Western group, and an Eastern group, and then there were two resident populations that came up as genetically distinct. That's my recollection of the Kestrel Genoscape. I think that yeah, I don't know that anything, like, again, sort of like Serena asked, or somebody asked at the beginning, like, if you're sequencing whole genomes, like at some point that is just the amount of mixing there is in kestrels. So I don't know what's going on in kestrels, but like within the West, based on the genomic signatures, it seems like there's a lot of mixing, meaning birds are moving pretty far from their natal site. I think, it's possible that because I think that we didn't use full whole genome, but I doubt that any more would come out of that. I'm not sure. What we are doing right now with kestrels is like adding the wintering ground component. So yeah, and just trying to understand the connections with the 5 different genetic groups that were able to identify.
Okay. Well, I hope that answers your question, Heather. Hopefully we'll have more information to come in the future. But yeah, I think that was the last question that we have for now. So first of all, I want to thank you, Kristen, for the amazing presentation and answering those questions and sharing all these different projects that you're working on. And of course, working towards, some similar goals that we have, right, related to bird conservation and education. So I wanted to ask you, is there anything that you want to share with everybody before we sign off as well?
Oh gosh, nobody's ever asked me that. No, I mean, just that with like an audience like this, if anybody has worked to contribute, samples, that we appreciate it, and none of this could be done without you, and we're just working really hard to make the most of all of the data all the time.
Yeah, and I'm sure it's just such a group effort, right? Like really a wide-ranging effort, and you can't do it without all the help that you're getting. So
no, and I also can't individually like communicate with all of the people that contribute, which is like frustrating. So it's great to have, a captive audience to be able to thank folks. So
all right, well, thanks again, and thank you everybody for tuning in tonight. Really appreciate it, and we hope to see you again at a future event. Have a great rest of your evening.